Good afternoon, everybody, and welcome to March's Global Health Compound Design meeting. And the speaker today is Mike Petrassi from Caliber. He joined Caliber about 2 years ago in 2016. But previous to that, he was at Novartis. Now, last month, Greg Landrum was talking about pipelines for processing results from high-throughput screens. And Mike is going to give a talk today based much more on the practical aspects of how you actually go about doing this. So I'd like to welcome him to give his talk on hit triaging and advancement to anti-Wolbachia lead compounds. The floor is yours. Mike, over to you.
Thank you, Carolyn. Hello, everyone. So I'll be describing the work that the team has done on our anti-Wolbachia lead compounds. And I'm going to preface this talk by saying that this is an active program. So I'm not going to be showing any discrete structures. I'm just going to be showing the process and strategies that the team has taken to try to advance and find an anti-Wolbachia lead compound. The other thing to note in context is that this program we've come a little late to the game, and that both Liverpool and AbbVie have some really advanced, amazing compounds in this. So what we're trying to do is come up with something that we think is something that can add to the overall portfolio. So just to give a quick introduction, we at Caliber's a young institute, and what we really see ourselves as a really good place to do neglected disease drug discovery. So it was founded in 2002 by Peter Schultz, and it's basically designed to take some of the critical path modalities and techniques that were used by Schultz and other workers back at Novartis to try to translate that in innovative way to develop compounds for neglected disease. So we reintegrated back into Scripps 2 years ago, and we're now leveraging that set of amazing biology and research and chemistry. We're collaborating now with the Global Health Drug Discovery Institute in Beijing. And one of the key things, and this is something that I hope to be able to stress the power of, is that the medicinal chemists here really partner with internal biologists and biologists at other companies to develop their small molecules. So if you think about working in a silo of doing just medchem or doing biology, that really doesn't work. We found it's much, much more advantageous to really have in-depth partnerships in all of our discussions and all of our strategies with internal biologists. So I'll describe here, when we're looking at a disease, one of the things that we wanted to address was elephantitis and river blindness. So Onchocerciasis is a very, very devastating disease where you have river blindness and elephantiasis. And it's mainly caused by worms that form nodules under the skin and in the lymph. So these worms have a significant lifespan, and there is a current therapeutic that's used. Some of them use ivermectin, some of them use doxycycline. So the idea that we're looking at is we would like to come up with a compound that can replace and have a real advantage to the patients over doxycycline that would result in sterilizing the worms that can cause these two very bad diseases. And one of the things that is associated with doxycycline is a long dosing time. So it's a typically 6-week regimen, and it is contraindicated in children and pregnant women. So we would like to come up with a compound that really addresses both of those liabilities and something that can be mass distributed in some resource limited countries. So that's really the goal of what we're trying to do. And the strategy that we're taking is we're going after Wolbachia. So Wolbachia is an intracellular bacteria, and it is symbiotic to these nematodes. It's required for viability and reproduction. So what we want to do is really kill the Wolbachia parasite and selectively sterilize the worm parasite to avoid Loa loa toxicity. So Loa loa is coendemic in a great deal of the areas where the Onchocerca is also. And if you look at this map right here of the O. bulbus and the Loa infections, what we would like to do is effectively sterilize these worms by killing the co-symbiotic bacterium, which allows these worms to live. We think that this will be a safer treatment, and we will avoid some of the encephalopathy that would be caused from also having a very rapid killing of the Loa parasite. So, if we take a step back and we try to describe what we want to do is we want to have our compound in patients treat river blindness and elephantiasis. And what we want to come up with is an IND. So what are the IND requirements? We have to have something that has got demonstrated in vitro activity, and what we deem as translatable in vivo efficacy. So what we want is something that hits the worms by sterilization, but doesn't kill them. And then what we would like to do is have something that has an advantage to patients. So over standard of care. So what I mean by that is if you're a doctor and you have a patient that comes in, you want to be able to give them this compound and say, this one will be able to be better than doxycycline because it's only dosed for 7 days. It's a safe compound that can be dosed to women and children. And ultimately, we can think of a stretch goal as something that would be a single dose. Now, when we're doing this, and this won't be a part of this talk, is that we need to demonstrate a clear safety index. Everyone knows about that. We need selectivity, exclusion, or reversibility. And for this particular program, we would like to see no effect on the Loa. The other thing that we will deal with when we get to later compounds is suitable physical properties so that these things can be formulated, these things can be produced, and these compounds can also be delivered in a way that's cost effective to resource limited countries. So, how can we think about going for this? Whenever you start with a disease, you need a starting point upon which to do medicinal chemistry. And if you have a great target for this disease, so for example, if you've got, INHA for tuberculosis or RNA polymerase for some of these other diseases, then you can go with a target. There is limited information on Wolbachia, so we decided to go with a phenotypic screen, where what you're doing is trying to develop assays that take advantage of the disease itself, and you can recapitulate in the laboratory preclinically. You're then unbiased towards what the exact target is, but what you hope to do is come up with multiple assays and multiple scaffolds that are able to recapitulate the disease preclinically. And this is the strategy that we chose. And what we realized is that we can take advantage of some of the ex vivo assays because we're working with a worm and some of the great in vivo animal models that are available through our collaborators. So we started with a high throughput screen. We did this in Drosophila cells because Wolbachia has difficulties and is very difficult to culture outside of cells as an intracellular bacteria. And we did this via high content imaging. So if you look here, basically you take Wolbachia infected cells, you grow them for 6 days, and then you stain for their RNA and total DNA and look at that via the imaging. And if you see here, you can see how this really looks in real time. And these are the plates upon which you want to look. We screened this a while ago, and we screened approximately 300,000 compounds. The Caliber library has grown to approximately 1 million compounds, and we've got an amazing repurposing library that I'm sure Arnab and some of the other groups here will be describing later. But what I wanted to show here is the primary assay data. So when we're doing this, what we need to do is we need to absolutely validate these assays when we know the known controls. And then we also need to know what our counter screens are down here, and we use the cell titer glow luminescence format to look at our counter screen in HEC293s, and then we're able to recapitulate what we hope is is representative of what's going on in the clinic with DOX and some of the other RNA inhibitors. So there's two images here, and the one on the left was the initial primary screen workflow when we got through about half of the screen. And what you see is that we got a reasonable hit rate. So we wanted a hit rate that wasn't, around 10%, and we didn't want a hit rate where we got no hit rates. The major thing we were really looking for was filtering it based on mammalian cytotoxicity, and then we were able to filter these hits as something that reconfirmed and met that criteria. We then took that and put it through our secondary assay, and that's where I'll be describing what we consider validated hits. So the secondary assay does worms in the nematode B. behangi, and it's a 3-day ex vivo assay. So what we needed to make sure is we've got a phenotypic assay. We don't have a specific target that we're looking at. So what we would like to do is ask the same question in multiple formats. Can our compounds kill Wolbachia in Drosophila, in behangi, in Onchocercia ultimately, and things like that? That's the approach that we took. Now the final primary and secondary screens are here. So again, it was around 300,000 compounds. We got a hit rate of around 0.7%, and we tested 1,400 of them in dose response. So you'll notice here there's a discrepancy. So we got 2,000 hits, but we only tested 1,400 of them in dose response. That is effectively associated with the logistics of doing high throughput screening. Not everything that you were able to do is re-available when you're looking at dose response. The compounds could not pass through QC. The compounds are not available from the original sources via internal at Caliber or the vendors that we use. And we try to address this by doing SAR by inventory in the later stage, but that's effectively something that always happens, is that we are not able to recapitulate and reproduce everything that is of the primary hit. Now, we then took a variety of compounds and we were able to bring that down to test 300 of them as powders. And we got a very nice confirmation rate of around 70%. So we were very happy with this. And then what we did was we were able to take a bunch of these compounds and filter them for the ex vivo assay. And 32 of those met our criteria in the secondary assay, which I'll be describing next. So our secondary assay relies on getting B. behangi from Athens at TRS Labs. So what we do is we culture the worms in these 24 well plates for 3 days. And then we then take the frozen Filaria and dissect the ovaries and stain the ovaries, because this is where we think is the most translatably relevant to having a anti-Wolbachia that'll affect the worms. So we do that with fish and DAPI, and you can see the real data down here. So this is the worms. This is just DMSO, and then you can see here, looking at the DTC stains, these are the ovaries, and this is the Wolbachia here, and this is the amount of Wolbachia elimination when you zoom in. So this is what happens when you put in doxycycline. If you'll focus your attention on the bottom boxes, you can see here there's a lot of Wolbachia still in this worm, whereas if you apply 1 micromolar doxycycline, you can see a significant amount of elimination. So this is graphed here, and what we used, because we had to set a criteria, is 75% Wolbachia elimination. We feel that is a reasonable starting point that we are using based on the fact that doxycycline works, and works hopefully clinically to cause, everyone who can take it right now's disease. So this is really the way, this is the workhorse, and this is the assay that we use primarily, and this is the cutoff that we use right here. So this is our program flow chart. This is the way we progressed our compounds. We do them within MedChem. We run our Wolbachia Drosophila assay. We use our Brugia behangi as our secondary screen, and we're always doing mammalian tox. We're very fortunate that last year the Global Health Drug Discovery Institute in Beijing formed, and they were able to join us, and together, both us and GHDDI have come together and really partnered on the MedChem. So the same size MedChem team that I have at Caliber, GHDDI was able to put together a team and join us as well. With these teams, I need to say that we always use WuXi. WuXi is a great partner for us, and particularly I use the Wuhan site. So with this, what we do is we have our criteria where we need 75% elimination in the Brugia screen, and then we obviously look through the ADME, and then we do mouse PK on this. Then we can look at rodent efficacy models, and then ultimately, when we get a good enough compound that is very efficacious, we'll look at it through rat and dog PK studies and see if we will have an appropriate profile to advance, and then ultimately we'll do some more advanced tier 2 ADME and safety profiling. So, as I said, and I'm going to show this a couple more times throughout the thing, but this is the most detail, and this is really the way the hits from the screen looked. So if you look at this graph right here, what you can see is not everything from the Drosophila assay translates to the worm assay, okay? And these are compounds that were very active in both assays. So we consider these validated hits. That does not mean that the Drosophila compounds are not something that we could advance, but what we did was we wanted to focus on the compounds that were active in both. We didn't want to be misled by a mechanism that we were not very attracted to. So again, this is really where we were at. And you can see these are known compounds that obviously we would be interested in, and some of them, those are the reference compounds. This was our original starting point hit that we ultimately taken into lead opt, and what you can see is all the other red compounds around there, those are compounds of the same class. So what you'll see is that these compounds have good potency down to sub 100 nanomolar, but this class also has things that are above 1 micromolar and compounds that are very, very potent against the worms, but not potent at all. So again, this is something that we need to deal with in lead opt. So it's imperative to go through all of this. And the issue that we were wondering is, why could a compound be active in the Drosophila cell, but not active in the Wolbachia? And here are some of the things and considerations that we thought about. And this is something that, you know, we are asking ourselves all the time. Whenever you're doing phenotypic screens, this is something that you need to address, is you've got multiple formats. What are the differences between the formats of the assays that you're looking at? And is there something that you need to worry about? And the first thing that we looked at is, obviously there's a difference between the two hosts. Is it a host factor targeting? If it is, then obviously we would not want to do anything with Drosophila. This is something that we struggled with the most, which would be point 2, is, are there specific morphology, and it's a different organism. So Onchocercia is different from Brugia, which is different from Loa Loa. All of these organisms may have different ways to deal with xenobiotics. So they may have different permeabilities, they may have different efflux, they may have different metabolism mechanisms that are all different. So this is something that's just a concern, and what we've done to try to address that I'll describe later. And then the other thing is, the Wolbachia genomes may vary. So this is something that we will have to address as well. And ultimately, the answer to that will be done in the clinic. So this is another way that we've analyzed our hits. So really, I use the 2 workhorse platforms of StarDrop and LiveDesign, and all of the information that the biologist will give me. So we take all of the information that we have and we load it into as many platforms as we can. And we do an initial filter, taking out the structural alerts. So the toxophores, which I'll describe in a little bit more, common pains, promiscuity in our HTS screens. So what I mean by that is, if we have compounds in the Caliber high throughput screening deck that hit in more than 30% of the screens, or they hit in screens that have common readouts, we have all of that information and we use that to filter out. So these are the main ways that we instantly apply to every single screen to do our triage. Then what we do after we've done that is we look through all of the scaffolds. We do match pair analysis. We do try to find things that really are what we're interested in, and all the parameters that we can look at to really decide on what would be an attractive set of scaffold and scaffolds or singletons to really take into the hit validation. What we found was that we found one scaffold, which is exemplified by these green dots out of all the blue, where it had decent lipophilic ligand efficiency. So that is one parameter that we use quite often when we are looking at these antibacterials, because we know that we're going to be adding hydrophobicity. These compounds typically can be very hydrophobic, and this is one thing that we'd like to optimize and have a good starting point right away. We did have several natural products that had excellent lipid, but because of their tractability of synthesis, we deprioritized them initially. So this is going back to what our role is in MedChem. And the first role that we're looking at right here, which is in the /discovery stage hit to lead, is the chemical matter, which is the chemical descriptors, the known toxicity for pains, and what profiling should we do? So if we do have a specific thing that comes out, how do we profile it and address that? So if you have an aniline, you have to do mutagenic aims, for instance. If you have a nitro group, you know you have to get rid of that and modify that some other way. So how do you really address that? If you have a carboxylic acid, you need to address, is it forming glucuronides and things like that. So this is all things that we need to look at. And then we need to be able to synthesize these compounds. So ultimately, that's going to preclude a lot of natural products. So filtering unattractive starting points from the HTS. Toxophores is something that we try to avoid right away. And this is something that I show all the time, is what I call our ultimate toxic compound on the right. So a lot of us will know a lot of these. You don't want to have a nitrogen chlorine bond. You don't want to have a nitro, aromatic nitro. You don't want to have an acetidine. You don't want to have alkylating agents. And some of these other, a variety of toxic compounds. I don't even remember where the reference from this compound was. It's just been floating around all of the MedChem groups that I've known. But this is another point that I wanted to bring up, is fenclosic acid. So if you look at each individual one of these components, this parachlorophenyl, thiophenes, while not great by itself, is fine. And then the acetic acid, all three of these moieties are safe individually. But if you look at the secondary metabolism, this compound caused jaundice in humans via structural rearrangement of the glucuronide acetate and was then able to label proteins. So you need to look at everything in context. And that's something that we try to do and incorporate in all of our starting points and HTS analysis. The other thing that we do, which I'm sure everyone knows a lot about, and I don't need to go through it in detail, is we filter out the pains. So we know all of these that are within our deck, and we can basically look at this associated with when we have, multiple hitters in the same type of screen assay format, and you get the flat SAR. So since we don't have a target, we can't do all the biophysical analysis to see if we're getting a one-to-one binding of our compound to our protein target. So this is a little more difficult to do in a phenotypic assay, and we have to be a little bit more stringent when we apply these metrics. The other thing that we look at quite often, and this is really where I try to be very strict when I'm doing my evaluation of a hit screen, is what I call drug likeness. So we all know of the Lipinski rules, but if you really start looking at some of the more constraints like ligand deficiency, sp3 fraction, and aromatic ring count, and start applying and putting those into the parameters that you use for the evaluation, this is something that we do all the time. And it's something that I think is not stressed enough, that you need to really try to maximize putting in a high sp3 fraction and a good aromatic ring count while keeping the constraints and complexity to a reasonable amount. So now I'm going to get to the data. That was the description of how we did our initial work. And now what we did was we had 12 hits from the HTS screen that represented, depending on how you decided to call it, a variety of scaffolds. So we then took them and we were able to resynthesize what I considered 5 separate scaffolds during our hit evaluation. And we would then do the standard synthesize around 20 to 30 intermediates if we were able to, and then get 20 or 30 analogs by SAR by inventory, either from our vendors, our internal sources, or some of our partners. And if the initial SAR studies did not show a path forward, so you got a flat SAR, you wouldn't get reproduction after you resynthesized it, you could not get any traction with the improvements in potency or improvements in PK, then we effectively deprioritized that series. It didn't mean that we couldn't advance that series. It simply meant that it wasn't a series that we were going to prioritize. And we really looked at this in a 3-month period. So what I mean by that is these two, WB01 and 02, were quickly filtered out. They didn't recapitulate very well when we looked at the Wolbachia elimination and the Brugia. But these compounds did. But we weren't able to improve the potency. We did have a lot of METSTAB, and then we did find out that these were apoptosis inducers and IGFR inhibitors in mammalian cells. So all of these we had to put on hold. So that's just the way that we did our workflow. Now, we did find one scaffold, exemplified by WB07, which had an amide appendage to it, that had good potency in the Brugia. It had decent potency in the Drosophila. Unfortunately, it was unbelievably cleared very, very well in mouse. We didn't even bother to do human, and it wasn't very potent. We did see a slight amount of toxicity in our HEC-293s, but nothing really in our HEP-G2s. Now, this compound had terrible PK, and we were not able to initially look at it, but we then decided to look at it because the tractable amount of SAR was there. And from amides, we know how to do some quick hit to leads, which will be able to validate if the compounds are real or not. One of the hit to lead areas that was successful is taking the amide and just applying the simple isosteres of the oxidiazole. We're then able to dramatically increase the potency by appending one of the aromatic rings with a dichloro substituent. That was very good. We did pick up a little bit of tox, but we were able to also keep good clearance. Now, that clearance didn't translate into good PK for the simple reason that when we made the molecule so hydrophobic, we lost a lot of our permeability. So what we were then able to do is take WB11, and the initial starting hit, 07, that we were looking at was not able to achieve any exposure at a reasonable dose. And I hesitate to say 50 is a reasonable dose, but that is what we were able to look at. And we weren't able to get any time over the EC90 or IC90. Whereas the isostere compound, WB11, is able to, at a 20 mg per kg dose with some organics in the formulation, able to achieve time over the EC90 and over the IC90. For this presentation, the IC90 that I'm using is the Drosophila cells, and the EC90 is the Pahangi, the ex vivo worm assay. So you can see, now we've got what we consider a tool compound, which we can use to look at, is this scaffold and is this progression paradigm something that we want to take forward? So if you look at the next slide, what we always do is we interrogate what we are doing. And the simplest way we can do this is we can take compounds. We know that DOX works very well. It's got a good linearity in both the Drosophila cells and the Brugia model. So we have compounds here, like W01, that also works. Now, if you look at our tool compound, what you see is that we then put it into our rodent efficacy model, which is looking at smegmatis and how much Wolbachia is in there. So we dosed it 50 mg per kg for 11 days and compared that to dox. Now, 90% elimination, where you see the compound binning effectively into two regimes of elimination, is something we were excited about, that it validated the series. It showed reasonable efficacy compared to dox at a dose close to it, but you can see we definitely have room for improvement. So we considered this at this point a validated hit series that we were considering for lead optimization. So now when we go into our strategy, we're doing a phenotypic screen. What do? We want to know which screening paradigm really captures translatable biology of the infectious disease. So the strategy is very simple. We collaborate with the experts that are going on in this. And the two that we were able to collaborate with very quickly was UCSF and the University of Bonn. Then what we did was we asked ourselves, what are the real concerns we have with our series? The number one is phenotypic hit. And is this MOA the best? We don't know that. Then the compound itself, the compound series. We've got very tractable SAR with good potency. We're able to see some in vivo efficacy right now, which is good, but it's not something that blows us away. It's not unbelievably impressive. So we ask ourselves, if we want to get a compound, what do we need? We need a compelling in vitro data package. So we need to not hit any of the mammalian targets, and we need to have good selectivity of the anti-targets that would give us an unsafe compound. We need to be able to modulate Wolbachia in multiple formats. Effectively, we ultimately want to go to onco, but what we want to have is something that's very robust. So we need to do this in multiple formats. That'll build the most confidence in our program that we will have a therapeutic that will work in humans, because humans and the parasites and the Wolbachia that will be in the clinic and in the field will be heterogeneous. So what we want to do is come up with a compelling compound that has robust activity in vivo. So that's really what we're looking at. What we always want to do is make something that is the safest as possible. That's paramount to what we do. And the way that we're addressing robust efficacy is, again, looking at it in multiple formats, both in mouse and in GERDs. So the strategy that we took towards that is we combined with the GHDDI. So they're a new organization that's looking especially at diseases of the neglected world and in China, and they were able to collaborate with us on MedChem. So what we did was we took WB11, we did our optimization, and at the same time, we were doing our core modification and subseries generation. So what I mean by that is we were optimizing all three parts of the molecule. Our one would be represented by the oxadiazole down here. And we were looking at both the original amides, the oxadiazoles, and we're able to find some aliphatic groups as well. Then what we were able to do is we tested the compounds in parallel in efficacy at the University of Bonn. The reason why we tested in efficacy is because the ultimate readout of the assay is a couple months. So that is just a function of how it works. Then what we wanted to do was profile and learn everything we could about 422W-11, and then ultimately take all that information and combine it into the right candidate. So this is what we wanted to do. These are all the liabilities in the standard way that we profile WB11, and we started along a lot of these advanced lead profiling. This is just a list of the way that we do things, and we've generated most of this data, not necessarily on 11, but some of the more advanced compounds as well. Then I wanted to show, this is an example. So this is just 1 week of how we are doing things later in lead opt. So this was last year. This was the end of July. And what you can see is that here's doxycycline. And you can see, again, it gets to about 75% at 3 micromolar. Whereas a lot of our compounds that we're looking at here, you can see even below 100 nanomolar down here, we're able to achieve significant amounts of Wolbachia elimination in the Brugia. So this is where we are able to take compounds and we're able to test them in parallel all at the University of Bonn. And what you can see, the original hit, or what I would call hit to lead validating compounds, it does eliminate, but it does have some regimes, and that's not great, and that's signified by the PK. We were then able to take a variety of compounds from different subseries and put them in vivo. And what you can see is two of these compounds dramatically stand out. They group very nicely in their elimination. They've got very well-defined efficacy that looks very potent. And we're starting to get to attractive doses, down to 30 mg per kg BID. So what we were able to learn, and we tested and wanted to know ourselves, was is plasma exposure a surrogate for what's going on in the animal, in the Lido, to look at and guide our optimization. So what we did was we took a couple compounds that did have limited exposure over the worm ex vivo EC90, but were very efficacious in the Drosophila cell. And you can see here, these compounds effectively did not work. So what that told us is that our hypothesis of using the worm EC90 is really what we wanted to use to drive MedChem. So the other thing that we needed to do is we needed to address the CYP P450 and the selectivity of killing Wolbachia versus having an effect in mammalian cells. So you can see for both of these, we were able to look at and be able to drive this, and we're able to use both StarDrop and LiveDesign to able to guide us in our decisions of what to make and what not to make. An example of this, and I'm using the date paradigm here, is one of the ways that we're able to advance one of our compounds is we're able to take the trifluoromethyl group and put it to the pentafluorothiol group. And what you can see here is that had a dramatic effect on CYP1A2, which is one of our liabilities. So this is our progression scheme upon the way that we've done that. And this doesn't always work out exactly the way you want when you combine things. So we were able to get compounds which had greater than 50 micromolar on 1A2, but not everyone was an improved compound. For example, this compound was very good and very potent, but it had inferior PK to 490. So it's not something that we were looking at as well. So this is a snapshot of really what the progression that we did was. We took our original hit, which had decent potency, moderate to good human liver microsome stability, and we were able to maintain that and increase that in some of our compounds in 417. Potency, we were able to pretty much always achieve because we were able to know from StarDrop and LiveDesign really what parameters we were able to do. We had little QSAR models that work quite well. And CYP1A2 we were able to address. So now what I'll show you is a little more information on 490 and 417, where we've got good compounds that we're very happy with. We did look at a variety of these two series, where we took 625 and we put it on hold for the simple reason that it's a little bit tight on hERG, and we're looking at 715 as well. So this is what I mean by when we look at the EC90, and what you can see is that 490, which has the pendant pentafluorothiol ether, it looks very good. It maintains significant multiples over the EC90 for 24 hours after a single dose. So this is just at 20 mg per kg, and we're very happy with this type of paradigm. It does address CYP1A2 a little bit, but it does pick up a little bit of hERG, which we'll address, and I'll get to that in the point later. So this is some of our more recent data generated by Bonn, and what we would like to achieve is something less than 7-day dosing. And I can tell you right now, the data that I'm most excited about is looking at 490. You can see here, we achieve, while not as good as doing 30 mpk QD for 490, if we simply dose it on day 1 and day 7 of a 14-day model, what you can see is that we do have a very attractive ability to deplete Wolbachia levels from Lido. So this is something that we're very excited about. And the GERD model is considered a more translatable model of this, and that's where we want to compare to doxycycline. So if you'll look here, what you can see is that we do not have effect on sigmoidus in the direct early killing of the microflora in the GERD model. So the compounds don't really do anything. But when you take one of our compounds, 715, what you can see is a sterilization. So this is something that we're very excited about. This compares well to doxycycline, which is a standard of care, and has been taken out to 18 weeks by our collaborators at Bonn. So we're very excited by 490 and 417's profile in the Wolbachia levels in these in this GERD model. So our future plans are to continue to optimize our potency and efficacy of these compounds, and consider combining our favorable properties into one candidate molecule. So we are considering both 490 and 417 as compounds to advance, and we are doing the standard pre-IND profiling right now. This is more of a summary, which I hope I was able to capture throughout the talk. And really what I would like to say is that we started from an HTS screen, which is an idea to deplete Wolbachia from the ovaries of Brugia Pahangi and from Drosophila cells. We were then able to use this Pahangi model to really drive our medchem and be able to absolutely go down in these directions to come up with compounds that were much, much more potent in our worm model, and then apply that in vivo. After we've applied that in vivo, then what we're trying to do is come up with compounds in dosing paradigms like 490 with a once per week dosing that will be very attractive to resource limited countries and be a good alternative for physicians to apply as opposed to doxycycline when the parameters would have a benefit to patients. So the last thing I needed to show is the acknowledgement slide, and Case McNamara is the lead biologist on this project, and Melina Bukowski is the person who effectively does all the biology work. Reza Shiroti was a postdoc in my group who was really leading this. The screen was done, led by Mitch, Hung, and Emily, who were amazing at this. Megan makes sure all the compounds go where they need to go. The Maxillary Disease Accelerator, funded by the Gates Foundation, has been instrumental in enabling us to get this done. And this has been a MedChem collaboration led by Jinjae Chu at GHDDI. And a lot of this MedChem was enabled by all of the synthesis that's really been led by Duan in Wuhan at WuXi. None of this would have been able to be done without our collaborators, Friedrich, UC Santa Cruz, and especially Mark and Akam at the University of Bonn. And again, none of this would have been possible at all without the funding and support of the Gates Foundation. Thank you, everyone, and I'll be happy to answer any questions.
Thank you, Mike. That was a great talk. If you'd like to ask any questions, then either raise your hand or just type them into the box and I'll pass them on. No microphone, says Chris Swain. Have you tried your compounds in combination with DOX or ivermectin?
So we're doing combination studies now. We've done that ex vivo and in vitro, but those become complicated to do in vivo. So we've not done them in vivo. They are additive in vitro, but they do not synergize.
I was going to ask you about when you put up your Lippy slide, you had some fabulous natural products there. Could you truly not have followed any of those as leads?
No, we absolutely could have followed those as leads. The issue that we had was twofold. It was logistical in that, one, there was a natural product already from AbbVie that is much, much more advanced from us. And we did not want to follow down a road that would have the potential similar limitations that the AbbVie compound would have that are associated with natural products. So complexity of synthesis, cost of goods, a dramatic way to a longer road uphill to optimize PK and do SAR. So those are some of the things that we strategically decided not to do as a strategy decision. But we absolutely could have followed up the natural products if that was something that we decided to go down.
Oh, that's really interesting. Thanks. Got any other questions in the audience?
Caroline, it's Mark here. I had a question. Sorry to anybody that wonders why they can't make themselves heard. I'm down as an organizer. That's why I can unmute myself and ask a question. I did type it in the chat box, but it probably, for some reason, as an organizer, I couldn't ask a question in the question box. Anyway, the question is about the PK/PD, and obviously you mentioned needing to get above EC90. I just wondered how successfully you'd been able to tell what it is in terms of, do you need time above EC90? Do you need some multiples at a certain time point? Do you need a large number of multiples at Cmax over EC90? And how you've been, to what extent you've been able to tell those apart?
So the short answer is we don't have an absolutely rigorous way, and by way I mean number of slots and resources to be able to answer that. What we did was we took one of the earlier compounds and we did a full dose titration, and it's one of the leads that is a precursor to 490, one of our last compounds. And what we were able to see is that it is trough level driven, and that you need to stay over the EC90 as much as you can. So you need to maintain levels above. It didn't appear to be Cmax driven. We tried some different dosing paradigms, and that was not effective. But the absolute numbers for multiples above EC90, we're still dose titrating to find the lowest efficacious exposure of our two most advanced compounds to try to answer that in the context of those questions. We know we want to stay at least 2 or 3-fold above the EC90 at trough level, but a rigorous answer to that we don't have yet, and I hope to within the next few months.
Yeah, great. Thank you.
Okay, we have another question here from David Hong, who says, I saw the formulation used in the in vivo PK and PD studies. 40% beta-cyclodextrin is quite unusual. Any particular reason for that? Can that translate into into the clinic easily?
Sure. So I'll answer the second question first. I would not try to advance a compound that required 40% cyclodextrin into human studies. So we used that formulation for a logistical concern. The compounds that we're advancing do not have the greatest physiochemical properties across the entire series. So because it's difficult to match exposures and we wanted to match the vehicle of each group when we were doing our initial evaluations, what we wanted to do was have one formulation that we could use as a control group to put a variety of compounds in to test hypotheses. So that's why we stuck to the 40% beta-cyclodextrin, because it was able to formulate well and we were able to get good exposure across a wide variety of compounds in these series and subseries. To address the advancement, what we did is, after we have a compound that we're very interested in, we would go back, do matching PK in, for example, MC-tween or CMC. So we would use very translatable, very benign formulations to match PK and then do dose multiples and make sure that we can get a compound in a simple suspension formulation using something like MC-tween to be able to get dose multiples, which would enable us to have confidence that we can do rat, dog, high dose PK and be able to achieve multiples so that we can do the appropriate safety studies in both rat and dog.
Okay, I've got one last question about your panes. The panes in your deck, do they differ significantly from the ones in the public set?
No, we basically just take the public set, and then when we have particular ones that we've seen from our screen, we add those as well.
Okay. I just want
every HDS deck has their own set.
Yep.
We add those as well.
Okay. Great. Any more questions? This is your last chance. No? Okay. Well, I think that it's probably time to round up Mike's presentation. Thank you very much. That was a great story and a really, really clear description of how you took the project through. And I'd like to wish you the best of luck with your clinical candidates. It looks like you're doing a great job there. And interesting to see that this was such a great collaboration, a really international collaboration. I can also make an announce that next month's talk will be given by our own Mark Gardner, and will follow on from the concern about being able to predict PK. So he's going to talk about PK Solver, which is a free tool that you can use to analyze PK data and derive PK parameters, and it complements the Gates-funded PK tool for analysis and prediction. It's an Excel add-in, and you can use it to predict things such as half-life, volume area under a curve. The original article was cited over 500 times, and he will describe some use studies. And that will be on the Wednesday, the 18th of April. So look forward to seeing you all then as well. And finally, thanks again to Mike. Enjoy the rest of your day.